Now I want to talk about the spermatogenesis. As you know, the spermatogenesis, it's a process of maturation of male gametes in the wall of seminiferous tubules. This is a section of the one seminiferous tubule in the human testis. Spermatogenesis involves a series of changes leading to conversion of spermatogonia into a spermatozoa. This is a spermatogonia, and finally we have a spermatozoa. In the male, the formation of the gametes, it means spermatozoa, take place only during the reproductive period, which begins in the age of puberty and continues even into old age. The duration time of the spermatogenesis take place around the 64 to 24 days. And if you look at the one of the seminiferous tubule on the microscope, like these pictures, we find that there are many cells of different size and shape. Most of these represent stage in the formation of spermatozoa, but some called Sertoli cell, this one, have only supporting functions. The various cell stages in spermatogenesis are spermatocytosis, meiosis, and spermiogenesis. In the spermatocytosis, in the process of conversion of spermatogonia to primary spermatocytes, this is a primary spermatocytes, this is the largest cells in the seminiferous tubule. It takes 16 days. It's by repeated mitotic divisions. As you remember, the PGC, when coming to the testes, the PGC give rise to a spermatogonia stem cell. We say that the spermatogonia is a kind of the stem cells. From these stem cells, a group of cells are found at regular intervals and are called type A spermatogonia. Marks beginning of spermatogenesis. It means that after the differentiation of PGC, we have a spermatogonia, and the spermatogonia divided to type A pale and A dark. The A dark is kind of the stem cells. It means that this kind has a characteristic of self-renewal and can making a type A pale. And in the last division of the A pale, the cell become the type of B spermatogonia. The type A spermatogonia are reserve cells, spatially dark kind of the type A. The type B is enlarged and undergo mitosis to form primary spermatocytes. And the primary spermatocytes enter to the mitotic division. It is process of conversion of the primary spermatocytes to secondary spermatocytes, and then finally we have a spermatid. This is the primary spermatocytes, and after the divisions, in the third mitotic division we have a secondary spermatocytes, and in the second mitotic finally we have a spermatid. The spermatid is a haploid cells, and the final stage in the process of spermiogenesis, we have a change in the shape and function without any change in the chromosome and number of the euploidy. In the spermiogenesis, what is the spermiogenesis? It's a process of metamorphosis of spermatid to spermatozoa and takes 24 days. The process by which a spermatid gradually change its shape to become a spermatozoa is called spermiogenesis. Its final stage in the maturation of spermatid to mature motile spermatozoa. Major events in spermiogenesis can divide it to three steps. One of them Golgi phase, to making the acrosomal cap. As you know, the acrosome contains many enzymes and secreted by Golgi apparatus, and these small vesicles join together and making an acrosome that contain many enzymes. This enzyme is needed for the fertilization and passing through the zona pellucida at the time of the fertilization. The acrosomal cap approximately cover the 2/3 surface of the nucleus of sperm, the dark blue in these pictures. This is acrosomal cap. The next stage in the spermiogenesis is a nucleus condensations. It means that in the nucleus condensations, the nucleus of spermatozoa be condensed because the one protein in the somatic cells that name is histones replaced by another protein that name is protamine. As a result of this exchange of between the histone and shift to protamine, the nucleus be condensed, and these events only happen in the sperm, is not happening in other cells. Okay, in the next and the last events during the spermiogenesis is tail formations. The tail formation is happen by a centriole. In the spermatid we have two centriole, proximal and distal. Distal centriole making a tail that has an axonomal structure, and the proximal centriole is remain in the neck, and this proximal is needed for the fertilization. Okay, in conclusion, in the spermiogenesis, as you see, we have some kind of the differentiation, and I divided this spermiogenesis in three steps: acrosomal cap formation, nucleus condensation, and tail formations. Here you can see that in the A pictures, the cross section from the testis of a newborn male embryo. You can see that in the testis only you see the primordial germ cells and Sertoli cells, and the B, as I told you, is a cross section from the testis of the adult persons. In this picture, it's in summary, you can see that the type A dark spermatogenesis that is differentiated from the PGC. As I told you, this is a stem cells and divided and making a pale, and a pale as a result of many mitosis division, making a type B and type D. Type B differentiated to primary spermatocyte, primary doing a first meiosis to making the secondary doing the second meiosis, and finally we have a 4 round spermatid, and as a result of the spermiogenesis we have finally some sperm, and the excess cytoplasmic from the round spermatid making a residual body is phagocytosis by the Sertoli cells, and finally we have a mature spermatozoa. This is again that same shape, spermiogenesis. In summary of spermiogenesis, in this picture you can see it. Spermiogenesis, the process, the Golgi apparatus to making the acrosomal cap, and finally the tail and the condensed nucleus. Sometimes we have some abnormality during the sperm formations, like 2 neck and 2 head, 2 tail, short tails. the large head and micro head. And in the clinic, we have some infertility, like abnormality in the shape. We say that the teratospermia, or the some abnormality in the morphology, in the shape, and something like this. And we need a normal shape for the fertilization. Severe male infertility, in which the ejaculated contains very few live sperm. We say that oligozoospermia, or even no live sperm, azoospermia, can be overcome using assisted reproductive techniques. We need the normal amount of sperm. In each ejaculation, we need at least 15 million sperm in each ejaculated semen, and more than 5% normal morphology is needed for the normal fertilization according to WHO manual. At puberty, now I want to talk about the first week of development, ovulation to implantation. First of all, I started from the ovarian cycle. At puberty, the female began to undergo regular monthly cycle. We say that the sexual cycle. This sexual cycle divided to 3 phases. The one of them is men's phase. It takes around 5 to 7 days. The follicular phase or proliferator phase is take around from the 6th day to the 14th days of the cycles. The last one is a secretory phase. Is started from the 15 days to the 28 days. For the purpose of better understanding the menstrual phase is described one by one. As you know, and around puberty, the anterior hypothalamic glands, or this picture shows that the pituitary gland, as you know, we divided anterior or adenohypophyse and posterior hypophyseal. And the anterior hypophyseal part secreted 2 important hormones, the 2 kind of gonadotropins, that name is FSH and LH. And FSH, follicle stimulating hormone, is responsible for the proliferative phase. As a result of the secretion of the FSH, and under the influence of the FSH, we have a growing of the follicle. In each cycle, around the 15 to 20 primordial follicle is started to growing. But during the time, at the normal condition, only one of these follicle reach full maturity, and only one oocyte is discharged. The other degenerated and become atrophic. In the next cycle, another group of primordial follicle is recruited, and again only one follicle reach maturity. Consequently, most follicles degenerated without ever reaching full maturity. At the same time, we have a maturity in the follicles. We have a change in the endometrium during this phase, as a result of secretion of estrogen that secreted by Graafian follicles. These Graafian follicles. Thus, proliferative phase of endometrium cycle coincidence with follicular phase of ovarian cycles. In conclusion, under the influence of estrogen during proliferative phase, epithelial cells are stimulated to grow and epithelialize an endometrial surface. thickness of endometrium is happen, and angiogenesis in the stratum functional layer of the endometrium is happen. Endometrial glands are stimulated to grow. To gland contain glycogen, but they are not secretory, and the ovulation occurs at the end of this phase. At the same time, it means in the time of the folliculogenesis, another event that's happened, tightening of the cervical mucosa occur, allow passage of sperm. Anterior lobe of the pituitary glands started to secretion of the LH, and at the mid-cycle there is an LH surge. This LH surge causes elevation concentration of maturating promoting factor MPF. This MPF causing oocyte to complete meiosis 1 and initiate meiosis 2. This LH surge causes stimulation production of the progesterone by follicular stromal cell luteinizations, and finally this LH causes follicular rupture and ovulations. And now about the ovulations. The shedding of ovum from the ovary is called ovulation. The ovarian follicle is at first very small compared to thickness of the cortex of ovary. Just before ovulation, the follicle may have diameter of 15 millimeters. The stroma and theca on this side of the follicle become very thin. A vascular area named is stigma appear of the most convex point of the follicle at the same time. During the ovulation process, there is a rupture of mature follicle and release secondary oocyte in the metaphase of second meiotic division. And high concentration of LH increase collagenase activity, resulting in digestion of collagen fiber around the ovary. Collagen fiber surrounding follicle. Prostaglandin level also increase in response to LH surge and cause local muscular contraction in the ovarian wall. Those contraction extrude the oocyte, which together with its surrounding granule cell from the region of the cumulus ovorus breaks free or ovulations. And when the follicle is ruptured, some of the cumulus ovorus cells then rearrange themselves around the zona pellucida, these points, to form the corona radiata. It means that the corona radiata is some of the cumulus cells that arrange around the oocytes. After the ovulations, granule cell remaining in the wall of the ruptured follicles, these granule cells, together with cells from the theca interna, these cells are vascularized by surrounding vessel under influence of LH. These cells develop a yellowish pigment and change into lutein cells, which form the corpus luteum and secrete estrogen and progesterone, especially progesterone. The progesterone together with some estrogen causes the uterine mucosa to enter the progestational or secretory stage in preparation for the implantation of the embryo. This one is a corpus luteum. The subsequent fate of the corpus luteum depends on whether the ovum is fertilized or not. If the ovum is not fertilized, the corpus luteum persists for about 14 days. During this period, it secretes progesterones. It remains relatively small and is called corpus luteum of menstruations. At the end of its function life, it degenerates and forms a mass of fibrous tissue called corpus albicans. Albicans means white body. But if the ovum is fertilized and pregnancy results, the corpus luteum persists for 3 to 4 months. This is larger than corpus luteum of menstruation and is called corpus luteum of pregnancy. This corpus luteum of pregnancy may occupy one-third to half the total volume of the ovary. The progesterone secreted by its essential for maintenance of pregnancy in the first few months. After the 4th month, the corpus luteum is no longer needed as the placenta begins to secrete progesterone. The generation of the corpus luteum in early months of pregnancy is prevented by human chorionic gonadotropin, HCG, secreted by the trophoblast cells of the developing embryo. The series of changes that begin with the formation of the ovarian follicle and end with the degeneration of the corpus luteum constitute what is called the ovarian cycle. An ovarian cycle has an average duration of 28 days, with ovulation occur at mid-cycle, around the 14 days. In this picture, you can see that the ovulation, this one is an ovary. In the ovary, you can see some follicle in the different stages. This is the ovulation. The ovary with some cumulus cells, we say that coronal radiata, is released into the uterine tube. And the time of the shortly before the ovulation, fimbriate of the uterine tubes sweep over the surface of ovary, and the tube itself begin to contract rhythmically. It's thought that the oocyte, surrounded by some granular cell, is carried in the tube by this sweeping movement of fimbriate and by motion of the cilia and the epithelial lining. Once the oocyte is in the uterine tube, it's propelled by peristaltic muscular contraction of tube and by cilia in the tubal mucosa, with the rate of the transport regulated by endocrine status during and after ovulation. In humans, the fertilized oocyte reaches the uterine tube in approximately 3 to 4 days.